Assalamu alaikum. Today is the second class of Mindray BC-2800, which is an quantitative autohematology analyzer. Today we are going to discuss about the reagents, controls and calibrators of hematology analyzer, which is a BC-2800 Mindray. Reagents are those which we use. To perform our test. Reagents are of many types and it is different from the different machines. As you know, we are using Mindray BC-2800. It means we should use the Mindray reagents for better performance of our machines and the better results of our results of our samples. If we use there is a point, and if we use the reagents which are provided by the third party, it may low your performance and it may be the variation in the results. Now the reagents are very important to perform the test, because if you don't have the reagents you can't perform the test if you have a machine only. Reagents are of many types. The reagents used in BC-2800 are: first one is the diluent. Diluent is basically, as the name indicates, that diluent is used for the dilution. As you know, when we sip the test in the machine never use the whole blood as all process to process the sample. It always make the dilution and then perform the test. Diluent have the following properties. Diluent first must dilute the blood samples, and the second property which diluent must have, it provide the environment between the cells and act like a plasma. The third one is the main property of diluent. It must provide the cell volume to the sample and act like a plasma to maintain the volume. The fourth one is it must be a conductive medium. It act as a conductive medium to help us to measure the WBCs and RBCs. First reagent is diluent. The second reagent we use in the BC-2800 is the lyse. As the name indicates, that the lyse is must a breakdown reagent which breaks something to make easy for the processing. Lyse is the breakdown. It break down the RBC's cell wall. It always break down a cell wall, release the hemoglobin from the cells. It always as you know, in RBC there is an hemoglobin. This is an RBC. Hemoglobin in the RBC. So it remove the cell wall of the RBC and release of hemoglobin from the cells. As you know, we also have to measure the hemoglobin. It reduces the size of cellular debris. As you know, the wall is damaged and there must be a cellular debris. It reduces the cellular debris to a level that does not interfere with the white blood cells counting. The second function is that it converts the hemoglobin which come from the RBC to a complex whose absorbance is determined the hemoglobin concentration. The third one we use as a reagent is rinse. Rinse is basically is formulated to rinse the bath and the metering tube and to provide the proper meniscus formation in the metering tubes and maintain it during the measurement cycle. It is basically a cleaning reagent. The fourth one is easy cleaner. Easy cleaner basically enzymatic. It is an enzymatic base isotonic agent, cleaning agent. It provide the cleaning property. It is basically a cleaning solution and wetting agent formulated to clean the fluid lines and bath. So it is also a cleaning agent. The probe cleaner is the last one, which is a alkaline based cleaning solution formulated to clean the fluid lines, apertures and the bath. The fourth thing we use, this is the first topic, which is the reagents, and now we are moving towards the controls. What are the controls? Controls basically controls and calibrator are used to verify accurate operation and calibrate the analyzer. Basically, controls verify the accurate operations which are performed by our machine, and calibrator which calibrate the enzyme and tell us. Calibration is basically the software that we provided to the machine which help which help in the processing of test, in the processing of results. Controls are basically commercially prepared from the whole blood and used to verify that the analyzer is functioning properly or not. If the controls are not in the limit that are provided by the Mindray, or we use any of the third party, or we made it from ourselves, if these are not in the range, it means that our machine is not working properly. Control, basically they are available in three ranges, which is the low, normal and high. Daily use of all three levels verify the operation of the analyzer and ensures that reliable results are obtained. And now it means that if we are performing all our tests, all our samples with three levels of controls, and our controls are in ranges, as low control is in the low range, normal in the normal range and high in the high range, it means that our machine is calibrated and all our results are 99% are correct. Now the last topic is we are going to cover is the calibrators. Calibrators are commercially prepared. They are also commercially prepared from the whole blood products, and it used to calibrate the analyzer. Calibrator are basically, most of the time, it are provided by the company, and we can also use the calibrators from the other company, but these are like the software which help the machine to perform and make results.
They can.